data analysis and experimental design for our project.
So for Adam's study, he worked with pluripotent stem cells at Colorado State University. During Adam's research, he observed two genetically identical cells in addition to iPS cells and studied their gene expression. He decided to grow fibroblast cells in a device called lab fibroblasts. They're easy to grow, that's why he worked with them. And during his research, he learned how to transform fibroblast cells into the iPS stem cells. He then found two genetically identical cell types, which were specialized fibroblast cells and unspecialized iPS cells. He measured the expression for each gene by observing the abundance of RNA in each gene and the different types of cells. After studying the functions of each cell, he concluded that each cell line will express genes that pertain to individual functions of the cell. I'm sorry, of the organism that it is needed to, like the proteins. I'm sorry, I messed that up. It's fine, but yeah, that pertains to individual functions.
Here is a graph of Adam's data that was recorded from his study. And the different colors represent the different types of the two different types of cells. On the x-axis is each of the measured genes, and on the y-axis is the amount of times that the gene was expressed in each of the different cell. You can see here the different results of how much the gene was expressed in each different cell, especially the ones that had the biggest difference of expression. The data shows how differently each gene he observed was expressed in each of the different iPS and human fibroblast cells. Each gene that is seen on the x-axis is seen in the different colors. Some genes were more expressed in the iPS cells, and they indicate what the function of the cell is, which is the same for the human fibroblast cells. For example, the SOX2 gene mainly provides instructions for the cell to make a protein that plays a role in making tissues and organs in embryonic development. This gene was expressed lower in the fibroblast cells than in the iPS cells, and this makes sense because the fibroblast cell is meant for maintaining the extracellular matrix, but the iPS cells are used for making new tissue. This can also be observed on the graph as well. There were other genes that were more expressed in iPS cells as well, such as the MYC, NANOG, and KLF4 genes. All of these genes' functions apply to the function of the iPS cell, which is why they are more expressed in those cells than in the fibroblast cells. The COL1A2 is an example of a gene that is highly expressed and found in the fibroblast cells compared to the iPS cells. The gene COL1A2 provides instructions for making part of a big molecule named type 1 collagen. Collagen is a very important structure of proteins that helps build tissues for the joints and connective tissues. The fibroblast cell would have more of this gene expressed because the function of a fibroblast shows that the gene is more needed because of how collagen helps in the matrix of the tissues. COL1A2 is not very expressed in iPS cells because it is not needed for that cell's specific function. There are also genes that are equally expressed in both of the cell types because they don't apply to the cell's main function but to maintaining the cell's health, such as the gene ACTB, which is a gene that provides the instructions for the cell to make the protein beta-actin, which is part of the actin family, and it plays a role in all of the cells because it helps the cell move, organizes the cell, and is involved in cell division and controls a lot of cell junction.
So our study is kind of a redirection of Adam's studies because we're going to look at how his study can apply to that of skin cancer patients. So we're going to specifically look at how the fibroblasts differ in a patient that has skin cancer versus a normal fibroblast, and we're going to look at how their gene expressions are how they change and what genes will affect like because so so we think that skin cancer causes cell division to increase, so we're interested in seeing the kind of genes that affects that, and we'll be looking at the same kinds of genes that Adam looked at in his study.
We were able to formulate a hypothesis that if we compare skin cancer fibroblasts to normal skin fibroblasts, the gene expression will increase in the gene that initiates mitosis, or the CDK1 gene in those cells that contain cancer, or that contain skin cancer. And our justification to that is because cancer is a very known disease, or it's known to increase cell division and reproduction. So we believe that that specific gene would be expressed more in the fibroblast of the skin cancer.
All right, for the experimental strategies, I'm just going to read the list. The first one is to collect normal and skin cancer cells from participants of similar age range, put them into controlled environments, same medium cell density, temperature, give time for samples to grow, check protein concentration for each gene type between skin cancer cells and fibroblasts, take 3 different protein counts of 3 cells from each group, skin cancer cells and normal cells, compare gene expression between cell groups, and share results.
So our predictive results is that the gene expression, specifically the CDK1 gene, sorry, I just like lost. So the one that initiates mitosis specifically will increase in the cells that are cancerous compared to the ones that are the normal cells. So if you look at the chart, the genes that have express the initiation of cell division, we are expected to see an increase in those in the cancerous cells. And then these are
done.